So welcome. Thank you for attending. Today's presentation, For Protons to Pictures to Party Tricks, is entertaining, interactive, and very informative. To introduce the presentation and today's speaker, I would like to introduce the moderator for today's session, Dr. Reggie Taylor. Dr. Taylor completed his PhD at Western University, where he developed magnetic resonance spectroscopy techniques on ultra-high field MRI scanners to measure neurotransmitters in schizophrenia and depression. That's a mouthful. He then completed a MITAX fellowship as a postdoc with Lawson Health Research Institute and intern with Siemens Canada. Now currently he's a physicist and scientist at the Brain Imaging Center at the Royal Institute of Mental Health Research, using their dual modality PETMRI system. He also holds an appointment here at Carleton as an adjunct research professor in the Department of Physics. Dr. Taylor, welcome.
Okay, yeah, thank you. And yeah, I'll get right to introducing Avery so we can get started on his talk. It's my pleasure to introduce Dr. Avery Berman, who completed his Bachelor of Science in Physics at the University of Victoria, then went on to complete a Master's in Medical Radiation Physics and a PhD in Biomedical Engineering, both under the supervision of Dr. Bruce Pike, who was a well-known and respected researcher in the field. His PhD thesis was on the development of advanced biophysical models to extract quantitative physiological parameters from the BOLD, or the blood oxygen level dependent signal. He then went on to complete a postdoctoral fellowship in radiology at Harvard Medical School, working at the Martino Center for Biomedical Imaging at MGH. This is where he focused on developing methods to improve the acquisition and modeling of high spatial resolution functional MRI at ultra-high magnetic field strengths, which would be 7 Tesla and up. He is a well-accomplished researcher already in his young career, and we're fortunate enough to have him working as an assistant professor in the Department of Physics at Carleton University and as a scientist at the Institute for Mental Health Research. Last year at the ISMRM, the main MRI conference in the field, Avery moderated a segment called the Artifact Game Show, where they put up an MRI artifact and the people in the stands have to try to guess what the artifact is. And it's always a good time. I can tell you it was a lot of fun to watch that presentation, and I look forward to hearing what he has to say today on his talk entitled From Proton to Picture to Party Tricks.
Okay, great. Thanks so much, Reggie, for that introduction. And thank you, Moira, as well, for the invitation, and same to the Office of the Dean of Science. I recently joined Carleton and moved to Ottawa just in January, so I'm quite green here in the city, and really my pleasure to get to interact with the community now. So this is my talk, From Proton to Picture to Party Tricks, and it's like my stream of consciousness overview of MRI and some of the more fun and entertaining applications that I've seen over the last decade or so, with a little bit of education sprinkled in. So let's get started with some education then. So MRI, for those who aren't aware, uses the nuclear magnetic resonance phenomenon, where nuclear is because we're actually deriving our signal from the atomic nuclei, primarily from the hydrogen atoms on the water molecule. And since our body is full of water, we have lots of signal sources. It's magnetic because we're actually measuring magnetic properties of the hydrogen nucleus, which is just one proton, so sometimes we'll say proton. And each proton is like a little bar magnet that interacts with the magnetic field in MRI. And then resonance, we're exploiting a resonance phenomenon, which is just a dependence on a specific frequency in NMR. And so one of the main phenomena as well is that when you put these protons or water molecules into an external magnetic field, like in an MRI scanner, they tend to precess or just rotate around this direction of the magnetic field. And the frequency at which they're precessing is directly proportional to that strength of the magnetic field. And this is relevant when we're actually trying to capture our images. And so how we actually capture those images, is we're using the NMR phenomenon, but we're adding some spatial encoding properties. So here we're seeing a cutout of an MRI scanner, and it has three main components. The first is it has a very strong magnetic field, which is just developed from a solenoid magnet. So just these coil windings going around the bore of the magnetic field, which you see often in everyday electronics, except in our case, we're using superconducting wires to achieve the really high magnetic fields that we need. It also uses a radio frequency system to transmit and receive signals from the body, just like your everyday antennas on your radio. And we then use these gradient coils to change the magnetic field as a function of the location in the MRI scanner. And unlike other imaging techniques like your camera even, we aren't directly imaging on a space by space basis. We're actually encoding in what we call Kspace, and I'll share more about that. So this is what Kspace looks like, and it's just related to our image space through a Fourier transform. So I'll go into some more detail on this later. Just to give you a sense for the strength of the magnetic field of an MRI scanner, it's roughly 100,000 times stronger than the Earth's magnetic field. And so we can see that the Earth itself is able to deflect the needle of a compass. So you can imagine then what might happen when you bring some magnetic objects near the MRI scanner. And so luckily, sometimes we get to do this. And so here's the case where scientists at Stanford University were decommissioning their MRI scanner. So they let the gas cylinder be introduced to the MRI scanner. So let's take a look. And maybe once more for good measure. So as we can see, the gas cylinder became a lethal projectile when going into the MRI field. However, MRI is very safe. But actually, this is the biggest safety concern, is the threat of projectiles around the MRI scanner. And this is why we do a really thorough screening of every patient or volunteer that goes into the MRI scanner to make sure that they have no metals on them. We'll even oftentimes have people change into scrubs, hospital scrubs, just to make sure that there's no loose change or anything left in pockets. Otherwise, MRI is very safe. And so in terms of the types of images we're able to generate, the contrast that we see is determined by a couple of different factors. So one of them is the properties of the tissue itself. And so there we're getting sensitivity to where the water actually is. So sometimes we'll call these images the proton density because there's just a single proton in the hydrogen nucleus. But we're also sensitive to the molecular environment. So is it more liquid? Is it more of a solid structure? And there's two different relaxation times, or two different parameters that dictate these sorts of images, and we call them T1 and T2. So here we're seeing images that are proton density weighted or T1 or T2 weighted. And we can actually tune our MRI scanner using different parameters of our what we call the pulse sequence. And so this is the timing and the power that we apply our radio frequency pulses, as well as when we encode our image and a few other parameters. And so with these tools in hand, we're then able to generate a staggering array of contrasts for instance, brain imaging as seen here. So we have some more conventional types of images up top, or contrasts up top, some becoming more conventional types of contrast here, which is really highlighting the veins throughout the brain, and some more exotic, which are primarily used for research at this stage, this sort of contrast, which is really emphasizing what we call white matter hyperintensities, which are structures often seen in neurological disorders and diseases like multiple sclerosis, for instance. But we're not always interested in imaging the water molecules. There's actually different species of hydrogen that aren't associated with water, that are associated with other molecules like glutamate or glutamine or choline. And so this is the field of magnetic resonance spectroscopy. So we can actually measure quantitatively what are the concentrations of these different metabolites, which are relevant for neuronal activity, as well as for imaging progression of tumors and cancer. So I'm a brain researcher. I'm biased in this sense, but you can also image many other structures like musculoskeletal systems. So I'm told this is a knee. We can also image the vascular system throughout the body. And so MRI can give us a great contrast with a type of technique called angiography. And because of just this really the versatility and the ability to image with such great contrast, all the soft tissues of the body, MRI has been really invaluable in diagnostic medical imaging. And it was awarded the Nobel Prize for Physiology or Medicine in 2003. And that award went to Sir Peter Mansfield and Paul Lauterbur, who are really instrumental to the development of MRI. So the story of MRI dates back actually to 1973. This is when Paul Lauterbur initially proposed and demonstrated MRI. It wasn't even in a body or even an animal or tissue, it was just a couple of vials of water, essentially, that he was imaging then. But he didn't actually call it MRI then, it had a different name. He called it zeugmatography, where Greek zeugma means that which is used for joining. And so because we're using multiple different magnetic fields in MRI, this was the term he came up with logically. But as we see, if we track the publications over essentially the first decade since the introduction of MRI, zeugmatography didn't quite take off as a name. But what did start to take off was nuclear magnetic resonance imaging. And so in the early 80s, people really started to adopt this as a name, and the techniques themselves also started to take off for medical imaging purposes. But also around this time was the Cold War. And as a result, the general population had a bit of a kind of nervous or anxiousness about the use of the word nuclear. So as a result, there was a PR move to drop nuclear from the title of this imaging technique, and it became known as magnetic resonance imaging. And as you can see, publications just started to take off with this name. And it's not because they made the name change, but it's just at this time, MRIs in general, the technology was taking off, but the publications all kind of followed suit with this PR stunt. And if we look forward to today, up till 2021, we're seeing really exponential growth in the use of MRI in both research and medicine. Okay, so at this point, we could take a break. This is a lot of hard work. I have a little quiz for the audience. I'm gonna try to pull up the chat box. So now I'm gonna put everyone else to work here a little bit. So this is a game called Guess the Fruit or Vegetable. So what we're gonna do is we're gonna watch slice by slice MRI images through a fruit or a vegetable. And once somebody in the audience thinks they know what the fruit or veggie is, please add your guess to the chat. And we'll try to allow maybe 15 to 30 seconds per item here. And just as a heads up, some slides will show multiples of a fruit or veggie. Example, there might be 3 mushrooms. So don't get confused that it's a weird structure. You're just looking at 3 mushrooms, but just guess, is it a mushroom or not? All right, so let's see, let's get started. Hopefully people can use the chat. Okay, yeah, and so put your guesses either to host and panelists or actually to everyone, I think. Message everyone if you can. The mushroom guess, I like that. Okay, well, it seems like relatively unanimous and seeing a little bit of dissent, but yes, this is indeed a pineapple. Great job, everyone. All right, moving on. Yeah, it looks like, again, it's unanimous this time. Ooh, a little bit of other guesses in there. Oh, the mushroom guesses, I like it. Yes, this is a kiwi fruit. This is a mushroom. No, 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 the rest of you got it right. This is corn. This is 3 corn on the cobs. Corns on the cob or corn on the cobs. 3 cobs of corn. Okay, results are a little mixed, but our first guesser got it, or at least the first on the chat that I saw. This is in fact an artichoke, not a cabbage. Cabbage, lettuce, artichoke, all good guesses here. Mushroom is also a valid answer. Okay, one more for now. Answers are much more mixed this time. I like that. Walnut. Hmm. Okay, I'm seeing a lot of the right answer here. This is in fact a cucumber. Will I show some? No, I don't think so. So yes, this is a cucumber. I would have accepted mushroom as well, but I didn't see that answer here. Okay, moving on to some education. I'm going to minimize the chat for now. So MRI is unlike most other imaging forms. So for instance, many of us have a digital camera in our phones, or we had just standalone digital cameras a couple decades ago. And in that case, we're using a lens to focus on the outside world onto an array of pixel detectors. And each area of the outside world that gets focused corresponds to an individual detector element that we then can display on a screen. And so we might have a million detectors, which then get translated to a million pixels on our display. MRI is quite different in that we can actually acquire all of our image data set on a single detector element. We can use more detector elements, but we do not need that. It's not necessary. So we can use a single detector coil to acquire a million, what we call voxels, which is volume elements, whereas pixels is picture elements, because we're acquiring our image in 3 dimensions. So it's got volume to it as well. And so I wanted to overview a little bit more about that. So I mentioned earlier that we're not actually measuring our image directly in image space like we are, say, a digital camera. We're imaging in what we call Kspace. What Kspace is actually the spatial frequency content of our image. And I think the best analogy is actually something many of us are familiar with, which is our music frequency decomposition of music or speech, whatever we're listening to. So oftentimes, we might have a visualizer on our music player that's showing dynamically changes in the bass frequencies all the way up to the treble frequencies. And these are changing, as we go. And what these are is, if we look at just a snapshot in time of, say, a music waveform, it's just a Fourier transform of our waveform there. So the Fourier transform provides a way of going between kind of our frequency space and our actual music space, time space, or in this case, we go going between K-space and our image space. So if we look at this frequency decomposition of our image, and we break it down into low frequency compartments and high frequency compartments, we can look at the low frequencies just by zeroing in on what's really in the center and ignoring everything on the outside. In this case, we get what looks like a low resolution or blurred version of our original image. And if we look at just what's in the high frequencies, we don't really see much of the image anymore, but what we're actually seeing is just the edges of our image. So these are areas that have rapid changes in contrast. So like we can see where the image goes from light to dark rapidly. That's where we're starting to see more structure in these high spatial frequencies. And by combining all of these, we get our original image. So what this means, though, is if we're really just interested in getting kind of the basic contrast of our image, we have to focus in on the center or these low frequencies of K-space. But the more we want to capture these fine details of anatomy, for instance, then we need to capture further and further out in our K-space. And this has consequences for how much time it might take to acquire our image. And so the way that we actually acquire our image is we're having to acquire each single point, or pixel, if you like, in Kspace one by one, essentially. And so we can do this by kind of raster scanning through line by line through Kspace. Now, this generally gives us our most kind of accurate representation of the object we're imaging, but it's quite long because we're having to repeat this process over and over and over, line by line. So there's other ways of speeding this up. We could do this all in a single shot and zigzag back and forth when we're reading out our image. So this is much faster, but it also has some cons to it as well. We can do other more exotic trajectories, like a spiral trajectory, if we like. And this is actually a rich area of research in MRIs, efficient ways to capture our Kspace or encode our Kspace with still accomplishing to get the contrast that we're interested in, or spatial resolution, or the temporal resolution. And so there's trade-offs in all of this in terms of how much time it takes to acquire our images, what spatial resolution we can then have, what contrast we might get, and as well, what sort of image imperfections or artifacts, as we call them, that we might be able to tolerate. And so with this arsenal of being able to generate our desired contrast using a specific type of image acquisition technique, we can do things like image the beating heart. So if we attach an external measurement to our patient that's measuring the cardiac cycle using an electrocardiogram, we can actually acquire data continuously in our patient and then bin that data according to where they were in the cardiac cycle and generate these beautiful images of the heart. Alternatively, if we capture images of the brain repeatedly over and over and over while a person's in the MRI scanner, we can actually have them do a task while they're there and measure changes in neuronal activity that result in changes in intensity differences in our MRI. And so what this is showing here are regions of the brain that, the bright yellow are regions of the brain that had increased neuronal activity in response to a task, and the blue regions are those that had decreased neuronal activity in response to this task. We can also image sibling rivalries if we like. So here we see twins in utero before, of course, being born. And so not only are we seeing the mother's internal organs in this case, we actually see exquisite detail in the internal organs of the fetus too. And this is a longer video, so I'll let it play out for a while. It's pretty amazing the sort of things that these twins get up to in there. And my greatest sympathies to mothers in general, pregnant mothers in general, but especially mothers of twins. This does not look like a comfortable situation whatsoever. So this one's rubbing its butt in its face. Yeah, really great video here. Also like to take this opportunity to point out that one of the best things as well as about MRI is that it uses non-ionizing radiation and no contrast agent injection at all. So it makes MRI one of the safest imaging modalities because of that, which is why we're able to image these fetuses here when they're already such a vulnerable population generally. So other things we can do is use MRI to do rapid imaging to study speech production, for instance, as maybe a linguist might want to do. So here's a video of that. I'll just let it run for about a minute. Now, unfortunately, MRI sounds nothing like the video on that last slide. It sounds much less pleasing to the ear. And so this is a medley of MRI sounds. So you might have experienced something like this yourself. Yeah, and in addition to it just not being very pleasing sound, it's also very loud. And so for this reason, we'll always give our subjects earplugs, and these are really, they reduce the noise level to really tolerable levels. But otherwise, without it, it's like you're in the front row of the rock concert and, right in front of the speaker. And so what's actually causing this is the switching of these gradients that we're using to produce the image encoding field. So we have these coils that we're running massive amounts of current through on and off. And so this is kind of a trace of what's going on. So on the top is the radio frequency field. It's not really a problem for us, but these are the waveforms that we're playing out to do the image encoding of the magnetic field gradients. And so they're rapidly switching their current on and off, on and off. And we have 3 axes because this way we can encode along X, Y, and Z. So arbitrary directions in 3D. And this is just a small snippet of the actual imaging time. And we're repeating this over and over and over with just very subtle changes to the image encoding itself. And when we look at the timing breakdown, so these gradients are turning on and off about 1 to 5 millisecond rate, which is a frequency then of 200 to 1000 hertz. The repetition time of these blocks are something like 5 to 200 milliseconds. So again, that's in the range of 5 to 200 hertz. And just a reminder, the human hearing range is 20 hertz to about 20 kilohertz. So all of these rapid switching of the gradients on and off are falling smack dab in our hearing range. And what's going on is when we turn the gradient coils on and off, it's displacing the hardware just slightly, but very rapidly. And this causes vibrations to propagate throughout the hardware, which then moves into the air and resulting in the sounds that we're able to hear. So not everybody, though, sees these MRI sounds as just noise or a nuisance that's loud. Charlotte Gainsbourg, who's the daughter of famous musician Serge Gainsbourg, has a different interpretation of it. And so she was actually able to incorporate it into some music of hers with the song here, ERM or IRM. So that's the MRI there. Not the drums, but the grating sound. Okay, yeah, also I love this song too, not just because it's using MRI sounds, but I think it's very catchy and cool song in general. IRM is not just MRI backwards, as I'm sure many in the audience here know. It's actually the acronym for MRI in French, so Imagerie par résonance magnétique. So we've seen now instances where we've used MRI to be able to visualize what's going on with a musician. And we've also seen musicians then take MRI and incorporate it into their own work. And so a group in Cleveland, Ohio was asking the question, well, what if we could do both? What if we could create music with the MRI, but also create images out of these? And music not just being these industrial sounds in this case. And so what they came up with was this. This is Cello Suite number 1 on MRI. And you might want to turn up the volume, it's a bit quieter. Okay, so I think that was our moment of Zen for the day. This was music all generated by the gradient coils on the MRI scanner. And the whole idea behind it was to try to improve patient comfort during an MRI examination. So we've heard what an MRI actually sounds like. You can imagine if you're a patient going into the scanner and you're anxious, and you're going into what's already a bit of a claustrophobic environment, that adding on those extra sounds to the experience is not going to be helpful and conducive, and you might actually have to withdraw from having the exam done. So by creating this more calming music, they would hope that they'd be able to retain some of these patients that otherwise wouldn't be able to get in the MRI scanner. And so the images that they generated look something like, sorry, let me back up. We'll get there. So what they did here in this study was actually they were able to take this cello waveform, which by the way was a recording from the famous cellist Yo-Yo Ma, and kind of reduce it to just the actual frequencies that were being played out at that time. And then they can translate that into what the gradient coils along X, Y, and Z would need to play out in order to recreate the music sounds themselves, and at the same time try to cover K-space to encode an image. And so now the images that they came up with look something like this. So not very useful whatsoever. However, this was just one image that they acquired throughout the entire song. They're acquiring several images over that time. Each of them a little different, but still unusable on their own. But if you look at the intensity coming from a single voxel, or a certain region of the brain in this case, over time, you can see that there's a lot of noise and fluctuations in that intensity, but there's also some overall or underlying structure. And so based on our knowledge of the physics of MRI and how different tissues have different properties, we could then create a database of signal types that we might expect to see, and then try to match the database based on the signal that we actually measured. And if we do that, then we can see, oh, hurrah, this is gray matter in the brain, and we know its relaxation times are this. This T1 is 1300 milliseconds, T2 is 110. So this now gives us really valuable information. And they call this technique MR fingerprinting, magnetic resonance fingerprinting, because just like in trying to determine the identity of somebody based on their fingerprint, which might be corrupted or impartial, but we can still identify them because it's unique. We're doing a similar thing with this database matching approach here, because it's a corrupted signal, but we still get enough information from it to identify the tissue type. And so then here are the sort of maps that they can then generate. So they're generating quantitative maps of these relaxation times, T1, T2, which on their own are very valuable. And you can use them to track changes in, say, tumor properties or regions of stroke, potentially, or hemorrhage. Or alternatively, wanted to generate images that are more conventional like, you can actually synthesize diagnostic images as well using these. So that's the bulk of the talk now for in terms of some of the interesting bits that I've come across. Just wanted to summarize what we did discuss today. So we went over the basic principles of MRI. So it uses nuclear magnetic resonance to give us image contrast, and then we use spatial encoding to generate the actual images. And this allows us to generate a range of contrast types that are good for imaging brains, knees, or corn on the cob. The spatial encoding can enable us to do fast or slow imaging, or high or low resolution imaging, again, used for, say, brain anatomy or fighting twins or vocalizing pop stars. The spatial encoding itself can be quite loud and noisy, but noise is in the eye of the beholder, as we saw from Charlotte Gainsbourg. We can also introduce randomness into our acquisition that can improve our quantitative MRI and provide a more pleasing environment for our patients. But now, more games. So everybody get your keyboard fingers ready. We're gonna guess a few more fruits and vegetables and then really gonna wrap up. Let me get the chat going too. Where is it? Okay, let's go. I'm seeing general consensus, but a bit of a mix. So yes, people are right. This is a tomato. Bubble tea. All right, generally consensus here. These are snap peas, indeed. I would have also accepted mushroom. Next. Oh, this is fun. I'm amazed with the audience on this one. I guess people eat a lot of pomegranate because this is indeed a pomegranate. I would have had a hard time guessing that. There must be a lot of cooks in the audience too, I'm guessing. Okay, slowing down a bit. And yes, this is lettuce. Don't ask me what type of lettuce. My guess would be romaine or curly green leaf, but I don't know. And this is the last one. Six. Mm. Wow, there's not much consensus here. I like that. This is a tricky one. For this one, I will not accept mushroom. Ooh. The guesses are still coming. So I've seen the right answer several times. Rebecca Thornhill, you were the first right answer. This is an eggplant. Sorry, next year I'll have to include the mushroom in this. Okay, so that's it for the fun stuff. Just wanted to do a few plugs here. So like I said, I'm from the physics department. We run programs from Bachelor of Science through Master's and PhD. And we're really focused on two main areas of research in the graduate department. We're doing particle physics research and medical physics research. In the particle physics domain, we have one of the largest theoretical groups working on particle phenomenology, largest in Canada, that is. And our experimental group really is unique that we're providing the whole spectrum from data analysis to instrumentation development for world-class facilities and experiments like SNOLAB here in Sudbury, and also CERN in Geneva. On the medical physics side, where I fall, we're using our knowledge of physics and applying it to medical imaging, radiation therapy, and radiobiology. More on medical physics. So Reggie and I are part of the Ottawa Medical Physics Institute, which is a network of roughly 40 medical physicists across the entire city. So it's not just people at Carleton. It's actually very few people at Carleton. There's only 5 of us, but it's mostly clinical scientists and physicists at hospitals, as well as scientists at national research laboratories like the NRC, as well as Health Canada. So it's really quite a broad group of people and quite unique to Ottawa as well, I'd say, because of the national labs. So we have a diversity and breadth then of topics that we're researching. And we offer master's and PhD degrees, of course, and on a range of projects that we can supervise. I also have an appointment at the Royal Hospital, where there we have a research dedicated MRI scanner. And so like Reggie had introduced, I work primarily on functional MRI and the development of new techniques for it. So this is a functional MRI experiment at one of the highest spatial resolutions that was performed. This is during my postdoctoral fellowship in Boston, but also doing investigation of ways we can image brain physiology. So things like blood flow and oxygen metabolism across the brain. Other researchers are working on imaging neurotransmitter function as it pertains then to neurodegenerative illness like Alzheimer's, which these images are showing, but also schizophrenia and addictions and PTSD. So the sort of things that Cliff Cassidy is looking at is he's looking at just very, very small regions of the brain just to look for very subtle changes in anatomical structure associated with neurotransmitters like dopamine system. And you can see that just these tiny little areas denoted by the yellow arrows here are bright in a healthy control, and are becoming darker in Alzheimer's disease. Now, the exact mechanisms for what contributes to this are still not entirely known, but they do have really strong correlations to disease progression in several sorts of disorders as well, like mental health disorders, as well as neurodegenerative disease. Reggie himself is an expert in MR spectroscopy, developing techniques to get at hard to measure neurotransmitters and metabolites with applications in major depressive disorder and schizophrenia, for example. But also one of the things that really makes us unique here at the Royal is that we have a combined PET and MRI scanner. So PET imaging is positron emission tomography, which is sensitive to actual molecules that will get taken up selectively by different target cells and organs. And then they will emit a small amount of radiation that we can then detect. And this provides very complementary information from the MRI. And so in this case, Professor Lowry Twelmanen has been measuring the physiological response to a new treatment type for depression. And they're actually looking at brain metabolism response after treatment on the PET scanner, just within minutes to hours of the treatment, and seeing significant changes in brain metabolism after this sort of treatment, and using the MRI itself to get the anatomical images for the underlay, as well as looking at some other aspects of brain activity. But this is still an ongoing study, so these are just preliminary results. In addition to brain imaging, we're also collaborating with the researchers from the Ottawa Hospital. Gerd Melkus is there doing bone imaging. In this case, this is studying overgrowth of bone in the hip joint. And so you can see that people with this disorder have an overgrowth of bone, which we're imaging with PET, which is with the particular tracer, which is sodium fluoride. It's sensitive to bone. And so that's what this kind of orange overlay is showing that there's some bone growth near the hip joint. And in a healthy control, we don't have that, that bright orange signal near the hip joint. At the same time, we're using a type of MRI technique that's sensitive to the cartilage around the socket here. And so we're seeing inflammation of the cartilage due to this overgrowth of bone. And so this MRI scanner that gives us the simultaneous information is really quite unique. There's only a few of them across the country. And so if people are interested in supporting mental health care or research at the Royal, there's always volunteers needed, as well as donations are always accepted. You can visit theroyal.ca. There's a get involved link. You could participate in research, study, or find other links of interest there. So I'd like to thank again the Office of the Dean of Science at Carleton. Reggie, thank you so much for moderating this, as well as colleagues of mine who made contributions to this talk. And thank you so much for attending. I'm open to questions and comments.
Okay, yeah, great job, Avery. That was a really good overview of what you can do with MRI, and also what someone could expect it to sound like if you were going in there. Sometimes I think it sounds like playing an 80s Nintendo game that just froze.
Yeah.
Yeah, exactly.
Yeah, and
Yeah.
after enough years of it, you know what scan's being done just by the sound of it too, right?
Yeah, for sure. And also, I think I'm gonna have to start calling it zoigmatography.
Yeah.
You know that. So yeah, if anyone has any questions, put them in the Q&A, and we'll get to them as in the order that they pop up. We'll get started with one that came up earlier. So you kind of touched on it in the talk. Is MRI dangerous? I mean, it's got nuclear in the word, so it must be. But what are the dangers of MRI?
Yeah, so the number one danger or threat in MRI is the threat of metals being brought into the scanner, so projectiles in that case. That's like I was saying, we're always trying to make sure that people don't have any metal on them or in them. Most medical metals like implants and things are MRI safe, so you can go in the MRI with that. But there are some types like shrapnel and things like that that you generally wouldn't want to have a participant go in the MRI for, especially not for a volunteer study. In emergency situations, it might be appropriate, but that's up to the radiologist to decide when to do that. But also, we just don't want to bring metallic objects near the MRI scanner in general, since they will get attracted to the center of the MRI, and it can be a dangerous projectile as a result. So a lot of medical equipment generally has to be there has to be an additional set made just for MRI safe, like gas cylinder. Oh, well, you wouldn't bring gas cylinders, but what sort of things like some carts that you might bring a patient into the MRI with are going to be MRI safe, different equipment. So that's number one. Number two is hearing as well. Because it is so loud, there could be significant hearing damage if we didn't give you protection. So again, we'll give people earplugs to bring the noise down to a reasonable level. I'd say number three, there's the potential, because we have these rapidly switching gradients, there's the potential that we could induce tiny electric currents or in the body itself. And so this can actually induce an action potential. So you can get like a little muscle spasm. That is generally just uncomfortable, but it can be uncomfortable enough that a person would withdraw from being in the MRI. Beyond that, you can also, if you have just the right kind of configuration, these loops can actually, the current can start to generate heat. And so things can heat up in the MRI. But these are things that are all generally preventable and avoidable by proper preparation of the subject, by just giving them the instructions of how to avoid all of these things. They're all avoidable.
Yeah, that covers just about all of them. So next question, someone was asking what kind of tasks can be used for functional MRI? What are commonly used?
So pretty much anything that you can think of that you can do while confined within this tiny bore. So we do a lot of visual-based tasks. So we have a mirror generally mounted above the person's face so that they can see what's going on outside the MRI scanner, and there we'll project something on a screen so they can see it by the mirror. And in that case, then we can present them lots of different sort of visual stimuli. We can have them watch movies in the MRI scanner. I was once asked but unable to participate in a study where they were going to actually have me smelling different things in the MRI scanner to see kind of olfactory processes. We can have people do subtle manual tasks as well. I've seen studies of people doing like driving a race car simulator. So lots of sort of perception tasks and decision-making tasks. A lot of psychology experiments in general can be translated to the MRI.
Yeah.
I guess another really important use of fMRI is in clinically is in localization of what we call eloquent cortex. This is areas of the brain that are involved with specific tasks like speech production and recognition. So many times if someone's having, say, a tumor removed, you don't want to be interfering with these parts of the brains because that's going to have a significant cost on their quality of life afterwards. So we can do these sort of mapping tasks in the MRI scanner to know to avoid these certain regions of the brain.
Okay, yeah, sounds good. So next question. There's a question about MRI sounds, what causes it, but I think you covered that after the question came out. But what about, in your opinion, what's the future of MRI? Where is it going? And maybe there's another question later on we can cover too. That what's the next generation of MRI scanners?
Cool. Yeah, so that's always a tough question. So you almost don't want to give an answer because it's just bound to be wrong. But I think, I mean, in terms of where we are today, a huge trend in MRI is reducing the time it takes to perform an MRI. Because right now it can take minutes, and we're blocking patients 15 to 30 minutes in the scanner, as compared to something like a CT scanner, which acquires your images in seconds. So there's a really big push to try to get more value out of our MRI because they're also so expensive. We want to be able to reduce the amount of time needed to acquire the relevant diagnostic information. So there's really huge pushes to be able to try to get the same diagnostic information just faster using just smarter acquisition and reconstruction techniques. So electrical engineers, so I was mentioning our role in physics, but a lot of people coming from electrical engineering backgrounds with signal processing backgrounds that are then making big innovations on how we can actually reconstruct our data. And then what's kind of the future MRI scanner? Is that what you're saying?
And next generation, what
next
generation?
Where's it going? Yeah, so it's going in two directions actually. Right now, there's a really big push to make MRI more accessible and more portable. So people, companies now are investing in making lower field strength MRI scanners that you could actually roll around through a hospital and bring right to a patient's room if you liked. And these were actually the field strengths we were using when MRI was first developed. I shouldn't say we, I probably wasn't even born then. But the images back then weren't nearly of the same quality as they are today because the hardware has just improved so much today, as well as the image reconstruction techniques that are used today. So we're actually revisiting what was old. It's now new again. On top of that, there's also a huge push on the research domain to move to ultra high field strengths, like Reggie and I did for our postdocs. Or maybe that was actually during your PhD, Reggie. But anyways, ultra high field strength, because this just gives us more signal to work with, so we can look at smaller and smaller details that otherwise are buried in the noise. So just by using these higher field strengths, we get higher signal to noise ratio, so we're able to see more. So right now on the fMRI side, I know there's like a one-off MRI scanner being built at Berkeley that's at 7 Tesla. So it's kind of like the conventional ultra high field scanner, but with all of the other hardware being optimized. And they're trying to get down to, I think, 500 micron voxel fMRI, or even smaller as their goal. And make that without even actually any really fancy or exotic pulse sequences. They're trying to get to that. So just amazing gradient hardware.
Yeah.
And receive arrays.
Yeah, for sure. Okay. So next question. Someone's asking if Kspace is sacred, and if there's any other space that we could do these reconstructions in. Maybe another way to put it would be, is there any other options other than say a Fourier transform to get these kind of images?
Yeah, so Kspace is not sacred. Actually, the original implementation of MRI did not use Fourier transform, did not acquire data in Kspace. It was using actually more similar reconstruction and acquisition techniques as CT scans, which is a projection reconstruction. And there, you don't really need to be thinking about things in terms of Kspace. There's also other acquisition techniques that are doing things in like the wavelet domain, sort of like JPEG image compression in a way. So there's certain features in on our images that you can represent in the wavelet domain as opposed to Kspace per se, to try to also make imaging more efficient and faster. And that's generally referred to as like sparse sampling MRI.
Yeah, it was a tougher question.
Yeah.
So yeah, there's lots of questions here. I don't know if we'll get through them all, but I'll just keep throwing them out there. That's a couple questions about going into the scanner with, say, titanium hips, or if you have dental work, can people still get scanners? And then another question related to that would be pacemaker. Can you go into the scanner with a pacemaker?
Yeah, so I mean, the first two are generally MRI safe and okay. They do tend to have a negative impact on image quality in and around the direct object you're trying to image. Say with dental implants, like many people have, say, braces, for instance, or permanent retainers. Those do cause some distortions in our MR images, but it's still safe to have the MRI. And similar sort of things with hip implants, we often see just kind of no signal around the implants or some distortion of the MRI as well. And then pacemakers, I'm not as up to speed on that. I know generally it's kind of verbatim, like or just verboten. I mean, we don't scan patients with pacemakers, but I'm not sure if there've been any updates to that recently. I think if there's any MR physicists who are doing clinical work on the call, maybe they would know better. Actually, they could put that in the chat.
I think they are developing MRI safe pacemakers. But that's something that you would have to confirm with the developer. And for, I know you know this, but for the sake of the question, typically around any MRI, there's a line. It's called the 5 Gauss line. And that line it's also called the pacemaker line, because that's the line that if you have a pacemaker, you can't cross without risking it turning off. So traditionally, no, you shouldn't scan people with pacemakers, but they are coming out with new ones.
Yeah, that was my impression. I just wasn't sure, how far behind the times I was.
Yeah, no, that was good. Next up, what's the? I'm just gonna keep going till I get stopped, I guess. Keep throwing the questions at you. So what are the differences between a PET scanner and an MRI scanner?
Yeah, so I think I've hopefully given a decent overview of the MRI itself. So there we're kind of measuring intrinsic signals from all the water molecules in the body. And we're using radio waves essentially to both get the signal and receive the signal from these water molecules. With PET, we always have to inject some sort of radioactive tracer. So the most common that you'll see is fluorodeoxyglucose, deoxyribose. Is that right? Fluorodeoxyglucose. Yep. Yeah. So FDG, and so this is essentially an analog of glucose with radioactive fluorine 18 associated with it. And this gets taken up into cells as if it's real glucose, and then it gets trapped in there. It can't be metabolized because it's just a little bit too heavy with fluorine 18. But it will give you an indication of metabolism throughout the body, anywhere. And so the idea here is because it's radioactive, it'll release positron. This is an anti-electron. And then with a half-life of roughly 2 hours, is it, or 14 hours? It's been a while. My PET knowledge
is 110.
110, thank you. Yeah, so 110 hour half-life.
Minutes.
And then minutes. Okay, so it's roughly 2 hours.
Yeah, about 2 hours. Yeah, you're right.
Yeah, okay.
That's right.
Yeah, it'll get released, and then it'll interact with one of the electrons that are just throughout all matter in your body. and then release two 511 keV photons. So it'll release two photons that you measure, and essentially at 180 degrees apart from each other. And we have a ring of detectors around the person that's measuring these, and we're looking for photons that come in at roughly the same time, at 180 degrees apart from each other. And from that, we can then reconstruct what's going on internally, where there's more of the FDG being taken up. And there's several other sort of tracers as well that we can use to get more specific types of biological information.
Yeah.
So the resolution is quite different as well. The spatial resolution that we can achieve with these is also quite different. It tends to be lower than MRI and takes a while to acquire all of the data because we're just waiting for this natural radioactive process and just passively imaging that way.
Yeah, I'm impressed with your knowledge of the PET physics. It's pretty good. And then I guess on that note, why would you want an MRI versus a CT?
Yeah, so MRI is really one of its main strengths is soft tissue contrast. So like we saw, I don't have any CT images, but if you guys to show you at least like a CT of the brain versus a CT and MRI of the brain, the contrast differences are just immediate and glaring. So MRI can provide really a lot of subtle detail changes in the soft tissue. So all the white matter, all the gray matter of the brain that you really aren't picking up so much with CT. Outside of that, there's also potential concerns with radiation. Generally for all diagnostic imaging though, we kind of set those aside because it's a question of what sort of treatment does the patient need? Do we need the CT scan? And generally we do. And so, I mean, CT is looking at really just X-ray attenuation is the main thing. And so it's looking at density of tissue, more or less, or electron density. So it's very different properties than what the MRI is looking at. The MRI is looking at not just the density of the tissue there, but also sort of what's the local environment, like how fluid or mobile are the molecules in that region. So we're getting just kind of what a CT gives to some extent with the density, but then a little more in terms of these different kind of molecular environment. There's a much larger range of contrast we can generate.
Yeah.
So CT is amazing for anatomy, especially in this sense. So like if you just want bulk anatomy and you also need an image in a snapshot, CT is perfect for that.
For sure. So yeah, you kind of already talked about RF damage and MRI and MRI safety. What about can someone go in there with a tattoo? There's lots of the discussion, or you hear that tattoos can't be, well, they can be dangerous to bring into a scanner. Is there any truth to that?
Yeah, there has been. I think generally most tattoos are safe, but there's certain types of inks that aren't. I think tattoos that date back to certain eras are not generally safe. There might be some metals in the inks. Again, I think this is the sort of thing that we're always screening for. Generally, the MRI technologist running the scanner will like really quiz you on the details of the tattoo. But generally, most tattoos are safe. Like if you got them in Ottawa, it's going to be safe. I think, the ones that the sailors were getting out at port in exotic destinations were sometimes more questionable.
Yeah.
Reggie, do you have something to add to that?
Well, yeah, if you're, if you got your tattoo in like a jail cell or something like that, might be a little less reliable. But for the most part, if you got it from a reliable source, you have no real concerns. Yeah. Okay, and rolling along. So there's a question about COVID masks, about whether and with them. Does that affect your fMRI image? And what about the metal strip there?
Yeah, I actually had a game on that exact topic that I held off on. But so in MRI, one of the things we also use for brain imaging is called diffusion imaging. And so I'm going to show an example in a minute here. where diffusion imaging is measuring kind of the bulk motion of water. And in the brain, we're using it to look at white matter pathways. These are the bundles that connect different regions of the brain. And so here's an image that actually,
Nice.
this is what we call the COVID era artifact, where someone had their mask on. And let me just show you some other examples of this. And so this was before the mask was on, and then this is after we took the mask off. And so we were able to get back the images that we desired. There's still some distortions in here, but those are just unavoidable, almost unavoidable, and nothing to do with the mask. And so the reason the mask is a problem is because there is this aluminum strip in that mask. And especially with this sort of manufacturing, like it's never just going to be a pure metal. There's all sorts of impurities that go into it. And so if we look at magnetic susceptibility, which is responsible for this distortion, water, which is similar to most tissues in the body, is say at -9. We'll just say arbitrary units, -9. Air is up at 0. So there's actually a big susceptibility difference between water and air. That's why we saw that distortion that I said we can't avoid. Aluminum's even further out. It's at +30 parts per million. And then we have all of these other impurities that we often see in metals, like stainless steel, or even little flecks of iron and things like that, that are probably in there causing such heavy distortions. So this is the COVID artifact that quite often we forget to take the person's mask off. And they're not magnetic, so they're not really a threat necessarily to our safety, but they do have severe consequences on image quality.
Yeah.
Great question. It's almost like you planted that for me.
That was a good answer. Had it all prepared. So there's a question on, is there a limitation on the type of neurotransmitters one can image? So I guess that's probably referring to Cliff's study, looking at the dopamine.
Well, it's probably Cliff as well as you to some extent, Reggie. I guess you're also doing some, different neurotransmitter imaging. So Reggie, can I field the question to you?
Sure. Yeah. So typically, you don't usually image neurotransmitters. To measure neurotransmitters, you either got to use something like PET or spectroscopy. So the one way that you can image, let's say you want to get a measurement of glutamate across the brain, it would be another type of sequence. It's called spectroscopic imaging. So pretty much what you do is normally you do spectroscopy over just one little voxel that's maybe 8 centimeters cubed in size, but you change up your sequence a bit, and you acquire that over the whole brain. It's a little bit tougher to do that, and you're a little bit more limited. But if you do that, then you can get measurements for a few different metabolites. So actually, thanks, Avery. So there's NAA, glutamate, creatine, choline, myoinositol. Those all play different roles in the brain. Glutamate is the main neurotransmitter that you would see there, and some people do image glutamate, but other than that, it's pretty difficult to image neurotransmitters.
Yeah, and I would say, I guess, about Cliff's study that they were looking at sort of the indirect consequences of changes of neurotransmitter levels. So again, I also said that the contrast mechanisms aren't fully understood yet. And,
that's right.
But it's related to dopamine system, I think, as well as just a general class of neurotransmitters.
And Cliff's study is kind of an exception where, like you're imaging a type of contrast that's sensitive to dopamine. So in a way, you are imaging, you're scanning for dopamine, but yeah, it's a little different.
Yeah, I kind of got lucky with that.
Yeah. Okay, we got still a few more questions, but
oh my goodness. Yeah, I'm going to say we're 15 minutes over time now. This is
sounds good.
Voice of the Universe. We could probably go until 5 o'clock. Those questions keep coming in.
Yeah, there's a ton of them. Yeah.
The presentation was incredible. I say one more question, and then we're going to say goodbye. How's that?
Yeah.
Have a good one to wrap up with, Reggie.
Well, let's see. How about just how long does an MRI scan take? If someone was going to go in there and get a scan, what can they expect? Like,
Yeah,
like-
so it depends if you're going in for a clinical scan, like say you had a knee injury and you need to, they want to look at any damage to your knee. I think they'll probably have you in the scanner for 15 minutes to 30 minutes, something like this. And they're not acquiring a single image over that time. They're acquiring generally like 4 to 8 different types of images over that time. So any single scan might run from 2 minutes to 8 minutes, depending on the level of detail that they're looking at. And so please hold still during that time, because if you move, we might have to reacquire that data set.
Yeah.
If you're going in for a research study, probably the longest we'd have somebody in the MRI scanner is for up to an hour and a half, which is really starting to push it. Most people have to have a washroom break in that time.
yeah.
So anybody who's considering putting their hand up as as a candidate for research, bear that in mind. Could be an hour and a half in a scanner.
Oh, every study is a little different.
I don't want to dissuade anyone, but
And there's often some remuneration as well.
there you go. All right. Well, I think that that'll conclude today. As I said, I could sit and listen for another hour. I'm sure everybody else feels the same. Thank you very much, Reggie and Avery, for great presentation today. It was, as promised, interactive and entertaining and so informative. Thank you very much again to everybody that's left. In the room, we'll be putting out the recording hopefully within the next couple of days. So view it again and share it out. Thank you.
Yeah, great. Sorry we weren't able to answer everyone's questions, but feel free to email me if you have any really pressing questions. I look forward to addressing them too then. So thanks so much for the invitation. Thanks again, Moira. Thanks, Reggie.
Thank you very much. Bye-bye.
All right.